So in this new topic, we will be talking about the movement of proteins in and out of the cells and between the organelles within cells. And we will focus on a process known as vesicle trafficking, which is a vital process for the life of the eukaryotic cell. A typical eukaryotic cell has about 10,000 proteins in it. For cells to function properly, those proteins have to be in the right place, in the right organelle, in the right position in the cell. So for example, water-soluble hormone receptors have to be on the plasma membrane. The DNA-RNA polymerase needed to be in the nucleus. Hydrolases needed to be in the lysosomes, and so on. So eukaryotic cells have a very sophisticated ways for moving proteins to the right position in the cell. And we are going to talk about this in this chapter. So in this picture, what is shown is this is a 3D picture in this opening slide, shows a yellow endomembrane network in an yeast cell, and the red being the mitochondria and the blue being the nucleus. And we will appreciate the role of these endomembrane network, which is shown in this yellow color in this chapter. So in this part 1 of the module 2 or 2.1, we will only cover the section 13.1 and 13.2. So in the section 13.1, we will talk about the trafficking and targeting proteins across the ER membranes and introduce the signal hypothesis. We will discuss the role of signal sequences in the proteins, which are addresses embedded in the protein to determine where they are going to be delivered. So most of the proteins in the cell are synthesized on the cytosolic polyribosomes. And so there is a messenger RNA that is being bound to the ribosomes in the cytosol as is being translated. Once a protein is completely translated and released into the cytosol, it will remain in the cytosol unless there are some specific targeting sequences as shown as like red right here in that protein, and it will direct it to a different organelle. This is the signal-based targeting. So there might be a nuclear targeting sequence. It could be a peroxisome targeting sequence. There could be a mitochondrial targeting sequence or a chloroplast targeting sequences. On the other hand, there is a special class of protein known as the secretory proteins, and they are synthesized in a slightly different way. Their synthesis begins in the cytosolic free ribosomes as shown in here. But once synthesis begins, these ribosomes bound to the messenger RNA moves into the ER and synthesis is completed in the ER. Once this protein is in the ER, either in the lumen of the ER or in the membrane of the ER, it undergoes a number of translational modification and is then transported to the Golgi apparatus, where it is further modified and finally transported to the final destination, which can be either lysosomes or it could be the plasma membrane. If this protein is a plasma transmembrane protein, or it might be a protein that is secreted from the cell. This is known as protein sorting, or sometimes protein trafficking, or protein targeting, or vesicle-based trafficking, many different types of names. And in this topic, we are going to focus on the targeting, transport, and synthesis of these secretory proteins. So the nomenclature is a little unfortunate and confusing, because when I say secretory proteins, you are probably thinking of proteins that are secreted from the cell, and that is true. But there are proteins which are translated by code and transported into the ER, or any protein that is made in the ER, processed and in traffic out of the Golgi, is a part of the large family of secretory proteins. So do not get confused by the nomenclature that all secretory proteins are not secreted by the cell, but they are manufactured and processed through the endoplasmic reticulum and Golgi, and from there they can be transported into different organelles in the cell as well. Not all proteins that are targeted to the ER are actually secreted out of the cell, and that is the take-home message from here. Just remember that. So what I want to do in this first part is to focus on what happens in the first step, which is what happens in the endoplasmic reticulum. So the most eukaryotic cells have a very well-developed endoplasmic reticulum. I say most because some lower eukaryotics, although they display ER function, it is very difficult to visualize a traditional ER within the cell. but the endoplasmic reticulum is an enormous organelle. In higher eukaryotics, probably half of the cellular membrane is made up of the endoplasmic reticulum. It is a very large branched or network organelle. In this immunofluorescence image, you can see that in the lower magnification, the endoplasmic reticulum, which is stained with blue, practically takes up the entire cell. This dark region is the nucleus that is not stained, and you can appreciate the extensive network or branching of the organelle when you look at the immunofluorescence staining at a higher magnification. What is important to note that is the internal space of the endoplasmic reticulum, which is also known as the ER lumen, is actually one continuous space. So in the entire network where the entire ER lumen is connected. So if you were standing in one part of the ER, you could walk through the entire network and reach every branch of that network. So this is the idea that how big is that organelle is. So here are some transmission electron micrographs of the endoplasmic reticulum. Since the endoplasmic reticulum is very actively involved in synthesizing these secretory proteins, there are a lot of ribosomes. At any time, there are a lot of ribosomes bound to the outside of this organelle. So this gives the ER a rough appearance. So most of the ER, as the rough ER is shown here, most of the ER is of this type. You can see the lumen of the ER, and you can see the dark electron dense spots that are lining the lumen of the ER, and those are the ribosomes. There is a portion of the ER which is known as a smooth ER, which does not have any ribosomes bound. Here is an EM of the smooth ER. It is rarer than the rough ER, and is usually the site where vesicles bud from and they move into the Golgi apparatus. Most cells have a little bit of smooth ER. The smooth ER is responsible for making lipids, hormone, particularly the steroid-based hormone. The cells that are actively involved in secretion of hormones, for example, these are Leydig cells that secrete testosterone, which is a steroid hormones. They have a lot of smooth ER. So the cells whose function is to secrete substance such as hormones have a smoother ER more in a typical cell. So to give you an idea about the rough ER and the smooth ER, where they are located in relationship to each other, here is a three-dimensional representation of these organelles. So you can see that most of the ER is rough ER. You can see that the rough ER has the ribosomes bound that are involved in synthesizing these secretory proteins. The smooth ER is found here right in the middle. This is the region from which the vesicles actually bud on their way to the Golgi apparatus. This part is shaded, what is the green right here. These are the continuous ER lumens. So that's the rough representation of the ER in 3D. So in order to study the ER in vitro, you have to purify it from the cells. We have talked a little about the organelle purification. At first, it seems like a daunting task because the ER is so enormous. How can you possibly purify this organelle? What is really advantageous for the cell biologist, if they want to study the ER, is that when you homogenize a cell, what will happen is that the membrane of the ER becomes disrupted, but they reseal to form these small vesicles, which are known as microsomes. Is that they are authentic miniature versions of the ER and function exactly like ER. So the interior is biochemically equivalent to ER. And in cases of rough microsomes, you only have ribosomes that are bound to the outside and synthesizing the secretory proteins. And so these microsomes are very easy to isolate and purify, that they make possible to study the biochemical and biophysical reactions that take place in the ER. So here is a rough transmission EM of rough and smooth microsomes. You can easily see that good example of the rough microsome right here. And you can see that there is no ribosome attached to the outer surface compared to this. This is an example of your smooth microsome. So let's say you want to study this process of secretory protein synthesis. You need to separate the rough microsomes from the smooth. Because they differ in density, you can do this equilibrium density centrifugation. For example, on a sucrose gradient, so you can take the population of the rough and the smooth microsomes and layer them on top of the sucrose gradient and apply a centrifugal force. And these small organelles will now move through the gradient, and they will stop moving in the gradient where density equals to the density of the gradient. So you can get a nice region of that gradient that contains fairly pure rough microsomes and another region containing fairly pure smooth microsomes, because their density varies due to the attached ribosome and no attached ribosome. So of course, you are studying the secretory proteins, then you are interested in the rough microsomes population in this gradient, and you are going to collect these population. So exactly how do secretory proteins get directed to the endoplasmic reticulum? Remember, their synthesis begins on the cytosolic free ribosome. Then once a synthesis begins and goes for a little bit, then that polyribosome, which is messenger RNA bound by several ribosomes, gets directed to the ER. So how does this happen? Well, in the 1970s, it was proposed that it happens through some kind of signal sequence in the protein. So here is a sequence, for this example, is at the N-terminus. So right here in the N-terminus, you can see that there is a red signal. This short segment of amino acid directs the polyribosome to the endoplasmic reticulum, and as synthesis continues, that signal sequence is cleaved off by a signal peptidase. So the final mature protein, that small amino acid tag, is not present in the final mature protein. And you can see that they are separated. This is the signal sequence. This is a mature protein without that signal sequence, or the portion of the protein which was at the N-terminal. This was proposed by Günter Blobel in the 70s and the 80s, and this was called as a signal hypothesis. So here is one of the experiments that he did, the Günter Blobel, and to support the hypothesis, and there was some kind of signal sequence on the N-terminus of the secretory protein. So what he did was took the messenger RNA encoding the secretory protein and allowed in vitro translation in a test tube to occur in the presence and absence of microsome, either presence of microsome or absence of microsomes. And he resolved them by an SDA's page, or the polyacrylamide gel electrophoresis. So he run these two samples. One is the protein with the microsomes, and another the protein without any microsomes. What he observed was an absence of microsome. The proteins were larger than the predicted, but in the presence of the microsome, they were of the correct size. This is the experiment that supported the idea that the signal sequence or some part of the protein was cleaved off in the presence of microsome. So in absence of microsome, the protein have the signal sequence. That's the reason that they are higher in molecular weight. On the other hand, in presence of microsome, the signal sequence is cleaved off by the protease or the signal peptidase, so the protein is shorter. So that's why it migrated on the polyacrylamide gel a little bit more. So the molecular weight of this is less compared to that. So this is basically the overview of idea. Now, this was a very simple experiment by today's standard, not very sophisticated, but done with the technology at that time. Now, there is much more data and much more sophisticated data that supports the signal hypothesis. It is strange that it is still called a signal hypothesis today because there is so much data to support it. So it's really not a hypothesis anymore. It's a proven fact. For example, the protein sequencing of the N-terminal amino acid sequence shows the exact sequence of the signal peptides. They are at the beginning of synthesis and not there anymore after transporting into the microsomes. Mass spectrometry that we talked about in the first topic on the course has also lent support to the hypothesis. So the scientific community proved over and over the validity of the signal hypothesis. So Günter Blobel was awarded the 1999 Nobel Prize for the work which was done more than a decade ago. So this gives you an idea about the importance of the signals in the protein trafficking. So here is an experiment that demonstrates that translation and translocation of the proteins occur simultaneously. So two different ones, translation and translocation. Translation is from the mRNA to the synthesis of the protein, and translocation is the delivery of the protein to the ER, or here microsomes. So here the messenger RNA for the secretory protein is bound to the ribosome, and you allow the synthesis of this protein in absence of microsome. Then you add the microsome after protein synthesis, and you look to see if your protein is inside the microsome or outside the microsome. What you find is that the protein is outside the microsome, so once synthesis is completed, the protein cannot be inserted into the lumen of the microsome. On the other hand, if you allow the synthesis of the protein to go on in presence of the microsome, what you will find is that the proteins are inside the microsome. This demonstrates that the translocation must occur simultaneously with translation. So the question might be, how can you tell that these proteins are really found in the microsomes? So here, for example, if you're allowing the synthesis of a secretory protein in the presence of microsomes, you're assuming that it gets translocated in the lumen of the microsome. How do you know that this is the case experimentally? What kind of characteristic can you look for to show that the protein has been successfully inserted into the microsomal lumen? The first thing you can do is a protein gel electrophoresis or SDS polyacrylamide gel electrophoresis to look at the size of the protein. Because of these proteins have been inserted into the microsome, the end-terminal signal peptide or the signal sequences will be cleaved off by the signal peptidase. This is the experiment that you just saw was the first conducted by Günter Blobel. If the protein is inside the microsome, it will be smaller. If it is outside, it will be larger. So the other characteristic of the proteins that have been translocated into the microsome that will be resistant to proteases, or they will not be resistant to proteases in the presence of detergent. So let's take a look at that. Let's say you have allowed the synthesis of the secretory protein to go on in some kind of cells in the culture. You isolate the microsome, homogenizing in the cells. And as you can see right here, if you add protease to these isolated microsomes, they are protected by the microsomal membrane, and there is no degradation of the membrane by the addition of these proteases. So now what you can do, that you can add detergent to these microsomal fraction, and these detergents can disrupt the architecture of the membrane. We discussed this in the last topic. What will happen that a small pores or disruption will form in the membrane. Then subsequently, if you add protease, the proteases will enter the lumen and then will hydrolyze those proteins. So in absence of detergent, the proteins in the lumen of the microsome will be resistant to protease, whereas in the presence of detergent, these same proteins will not be resistant, but will be sensitive to hydrolysis by proteases. Another thing is that important part is that they also become glycosylated by the enzyme within the microsomes. And we'll talk about those in later parts. So let's take a look at a little bit more of the signal hypothesis. The signal peptide, which is the short amino acid sequence and sometimes called the signal sequence, or sometimes called the leader sequence, or sometimes called the start transfer sequence. These signal peptides are about 30 to 35 amino acid in length. The sequence itself is not well conserved, but there are some features that are conserved. There are usually a stretch of hydrophobic amino acids. Here they are in the green, and these hydrophobic regions are usually followed by a couple of negatively charged amino acids, which is shown in the blue, and one or more positively charged amino acid shown in red. So the sequence itself is not conserved, but some of the hydrophobicity and some of the positioning of the negatively or positively charged amino acids are conserved. So here is another example. This table actually shows a typical signal sequence for your reference to show the conserved role of hydrophobic and charged amino acid residues in protein targeting to reach their destination. So this is the sequence we just saw, but you can easily see that if there is another sequence like this, the protein is retained in the endoplasmic reticulum. So to send the proteins to mitochondria, you have this signal. To the nucleus, you have a nuclear signal, peroxisomal signal. So there are many of these charge residues in certain combinations that can form the signal sequences. This is just for your understanding, not needed for quiz or exam. So here is a bigger picture of the process. This is a secretory protein being synthesized on a cytosolic free ribosome. As the protein becomes extruded or exposed out of the ribosome, the signal sequence, which is purple right here, becomes exposed. That signal sequence then binds to a riboprotein complex known as the signal recognition particle, or SRP, a very, very large complex. SRP will bind the signal sequence and temporarily halts or terminates the translation. Again, temporarily. So the protein synthesis and translation is halted until the polyribosome is directed to the endoplasmic reticulum. It is the signal recognition particle that directs the polyribosome to the ER membrane, and it does so by interacting with the receptor on the ER membrane. Here you can see as an alpha beta, and this is known as the signal recognition particle receptor, or SRP receptor. As SRP and alpha beta interact with each other, it brings the polyribosome on the ER membrane. So this is what the signal recognition particle looks like. It is a very, very large riboprotein complex, which means that there is an RNA associated, and there are 5-6 other proteins also associated with this particle. Note that here, one protein known as P54 subunit of the protein, and this is the signal recognition protein, and these are some of the signal sequences that it can recognize. So here is a molecular structure of the bacterial FFH protein, which is an homolog of the P54 portion of, this is the P54, and this is the P54 homolog of the signal recognition particle, or SRP. So here is being shown that this is the hydrophobic cleft where the hydrophobic amino acid side chains bind, and this is the protein binding sequence to the SRP. On the right here is a structure known as FTSY, which is an archaeal homolog of the alpha subunit of the SRP, which was not shown in the previous picture. This is actually a dimer which is bound to these GTPs. So the GTP is needed to dimerize and activate this complex and the whole SRP. Once the function of the SRP is done, which is bringing the polyribosome to the endoplasmic reticulum, these GTPs are hydrolyzed to GDP, and these two subunits separate, and the SRP is again will be charged by adding another GTP. So that will be the cycle that the SRP goes through right here. Here it will be dissociated from the GTP. It will hydrolyze the GTP into GDP, and it will go back into the again by picking up GTP. We're going to take a look at that soon. So here is once the polyribosome binds to the ER membrane through the integration of the SRP and its receptor, translation can be initiated again. So remember that when SRP bound, it was halting the translation temporarily. Now, upon binding to the SRP receptor, this will start back again. So the signal recognition receptors are usually associated with a large transport complex, and this is known as a translocon. Translocon is the word. So what happens is that this ribosome, which is translating the secretory protein, becomes dissociated from the SRP and its receptor and becomes associated with a translocon, and the translocon opens, the synthesis is reinitiated, and you get co-translational translocation of the secretory protein. So as translation is being done in that process, same way the translocation of the protein is happening, which means that as the protein is extruded from the ribosome, it is actually being transported to the translocon, and the translocon will guide these protein into the lumen or wherever it need to go. So as we have been discussing, the signal sequence is cleaved by the signal peptidase, which is found in the lumen of the ER. What you end up with is the mature soluble secretory protein. So this is the signal sequence cleaved off by the signal peptidase, and this is your mature soluble secretory protein. So here is a classic example to show that Sec61 alpha is a translocon right here, Sec61 alpha, and this is from an yeast experiment. So this is known as a cross-linking experiment to prove that Sec61 alpha is a translocon, and it contacts the nascent secretory proteins as they pass through the translocon into the ER lumen. So to do this experiment, an in vitro selfie translational system was taken, and there were liposome containing only Sec61 complex, an artificial mRNA encoding only about N-terminal 70 amino acids of the secreted protein prolactin. Beside the mRNA lacks a stop codon so that the ribosome never dissociates. The mRNA contains one lysine codon near the middle of the sequence, and these are all artificially made, that whole mRNA. And the reaction mixture also contained lysyl tRNA modified with a light-activated cross-linking reagent attached to the lysine side chain. So in an in vitro cell-free system, the entire mRNA is translated, but stuck in the translocon because ribosome will not be able to release the protein without an mRNA stop codon. The in vitro translation was followed by an exposure to the intense light, which will covalently cross-link the nascent polypeptide right here, will covalently cross-link the nascent polypeptide chain to the nearby proteins in the translocon through this photoactivated lysine. And what happens here, that as the photoactivated lysine is bound here, isolation of the translocon shows the presence of this peptide. So the translocation is driven by the translational elongation of the protein, and the energy for this comes from the ATP used in the translation, which is pushing the protein through this translocon complex where it is Sec61. The translocon itself do not use or expend any energy to push this protein through its channel. So this is how the cross-linking experiment, that the binding of the translocon into the lumen of the protein that is being synthesized, proved that this is the proper theory or the proper process of the translocation through the translocon from the ribosome to the endoplasmic reticular lumen. So here is a structure of an archaeal Sec61 complex and the translocon preserves the high integrity, and it uses about an kind of like an hourglass shaped channel to the center of this pore. And without the incoming peptide, the channel is closed by a short helical plug, which is shown in the red. It is closed right here, and which then moves out of the channel during the protein translocation. So no protein, it stays here. If protein is coming in, the plug is moved, so this channel is open. So this is the first gating mechanism. Even when a polypeptide is actively translocating, there is a ring right here. These are the green ones. These are the green ring of isoleucine, which constrict the waist of the pore and forms a kind of like a gasket in the middle of the channel. And this seals the channel so that the small molecules cannot slip by or come into the lumen when the protein is translocated. So this is the second gating mechanism. So here is another rendition of this process. Here is a secretory protein being synthesized and is initiated in the cytosolic free ribosome. Here you have an exposure of the signal sequence which bind to the SRP and then dictates the ribosome to go to the SRP or directs the polyribosome to the ER membrane through the interaction with the SRP receptor. The ribosome becomes associated with the translocon and synthesis begins again. Here is the signal peptidase that is cleaving off the signal peptide, and synthesis continues until the properly folded soluble secretory protein form in the lumen is delivered. Now, the most soluble secretory proteins are made in this way. And what I mean by a soluble secretory protein is that it is not membrane-associated protein. So it is free in the lumen of the endoplasmic reticulum. Now, most are synthesized in this way, but as you can imagine, there's an exception as well. So in the yeast, many of the secretory proteins translocated into the ER after they are completely made into the cytosol. See, all our previous example that we are seeing that if it is a secretory protein, the ribosome need to be associated with the endoplasmic reticular membrane before the secretory protein can be synthesized or it starts synthesizing back again. But in yeast, that is not the case. The yeast display a process known as post-translation translocation. So translation is not completed in the cytosol, but then the protein is fed through a translocon into ER. Here is some evidence that there may be some soluble secretory proteins in higher eukaryotic cells like mammalian cells that are made in this way. It is rare, but there are some examples. So this is the way that how it looks like in the yeast. The secretory protein is completely made in the cytosol. It becomes associated with the same translocation that is used for the formation of majority of the secretory proteins that use SRP. But in this case, SRP and the SRP receptor are not used. It does have a signal sequence and it becomes associated with the translocation. It starts to move into the lumen of the ER. So you can imagine that there would be some risk of this, because the protein may slide back into the cytosol. So what we did, that when we were doing this in the previous experiment, here the ribosome sits on top of the translocation, and that's the reason that the protein which is being synthesized is pushed only in one direction. It cannot go the other direction. But what happens here, the backsliding is a possibility because there is no ribosome sitting on top of the translocation to stop this process. So what happens in this condition, the way yeast have solved this problem, is through this protein known as BIP, or binding protein, which is the ER resident protein and is very abundant in the lumen of the ER. So another protein complex in the ER known as a SEX63 complex. So as secretory protein is moving, as it's moving through the translocation, SEX63 facilitates the binding of the BIP or BIP to this secretory protein. This is an energy consuming reaction, so ATP is hydrolyzed. As the protein continues to move inside, right here, it is constantly being added with these ADPs over this process. So you can see that this is an ATP hydrolysis, and the ADPs are added on every 30 residues of this protein. So you can almost see that this is the only way that it can prevent this backsliding of this into the cytosol. So this protein now almost looks like a kind of a chain with beads on it, so that this prevents the backsliding of the protein. So you end up with a mature soluble protein, secretory protein, in the lumen of the ER in these yeast cells finally. So in the first part of this topic, we looked at the synthesis of soluble secretory proteins on ER ribosome or ER polyribosomes. Remember that soluble secretory proteins are those that are synthesized on ribosomes that are bound to the ER, and the entire protein is extruded into the lumen of the ER. It is not membrane associated at all. However, the membrane associated proteins, like the integral membrane proteins, are also synthesized by the secretory synthesis pathway. So it is really important to consider the synthesis of integral membrane proteins in this subject. Where I talked about the membrane proteins, you will remember that there are integral or transmembrane proteins and the lipid anchored proteins that are tightly associated with lipid bilayer. And that's pretty much the talk of this part will be. So let's talk about the synthesis of both groups of these proteins on the ER-bound polyribosome. So let's start with the transmembrane proteins. Previously, we talked about the transmembrane proteins when we were talking about the biological membranes. We talked about single-pass transmembrane proteins and multiple-pass transmembrane proteins. And remember that the single-pass transmembrane proteins are those, as their name suggests, pass through the membrane only once. They can have the majority of their protein on one side of the other, or they can be equally distributed. The portion of the protein that passes through the membrane is a stretch of hydrophobic amino acid sequences and about 20 to 25 amino acids that form an alpha helical structure. This is the most common way a transmembrane protein spans a biological membrane or the phospholipid bilayer. And then there is these multi-pass proteins. As the name suggests that they pass through the membrane multiple times. What we didn't talk about, that we talked about this group of proteins previously, is that this group of proteins can have different orientations. So for example, if you have the C terminus or the cytosolic side of a membrane, or they can have the N terminus on the cytosolic side of the membrane. So in the case of, say, insulin receptor, the most of the protein is in the ER lumen side or the extracellular space, because remember that the lumen will be facing exterior, and that's what we talked about, where the cytosolic portion will be cytosolic. It will remain the same. So make sure that you are clear about this concept. Whereas for the case of, say, cytochrome B5, most of the protein is on the cytosolic side. So you can appreciate that there could be different topologies or different arrangement or orientations of these single pass transmembrane proteins. And this is also true for multiple pass proteins as well. If they pass through the membrane equal number of times, likely like the GLUT1 transporter, which does have 12 transmembrane domains, then the N-terminal and the C-terminal sides will end up on the same side, whether it's cytosolic or lumen or exterior side. On the other hand, if they pass odd number of times, then the amino terminal and the C terminal will end up in two different areas. So here we have 12, 1, 2, 3, 4, 5, 6, and 7. So odd number of times, and the N terminal and the C terminal in the beta adrenergic receptors end up on two different sides of the membrane. So you can understand the importance, because if the cell has a device that different ways to synthesize these transmembrane proteins such that their orientation is correct in the membrane, because without the correct orientation, these proteins are not going to be functional. That's the most important part. So you can actually group these transmembrane proteins into several types based on the orientation. So here is the type 1 transmembrane proteins, and they have a small portion of their C-terminus on the cytosolic side, a transmembrane region, and the majority of the protein is found on the lumen of the ER or the lumen of the Golgi. Now, please recap from the last modules how the lumen of ER Golgi correspond to the exterior or the exoplasmic phase of the cell that we just talked about in the previous slide as well. Type 2 transmembrane proteins, on the other hand, right here, have the opposite orientation, where a small portion of the N-terminus is on the cytosolic face of the phospholipid bilayer. And the type 3 is similar to type 1 in its orientation, where the C-terminus is on the cytosolic face, except the majority of the protein is on the cytosolic face. There's another group of proteins that can be considered as a type 3 in a reverse orientation, where the tail of the protein or the C-terminus is inserted into the membrane. These proteins are known as the tail anchored, because C-terminus is the tail of the protein, and this is known as the tail anchored proteins. And then a type 4 are multiple transmembrane alpha helices or multipass membrane proteins, which can be further divided into type 4A, which is N and C terminal both end up in the same side of the ER membrane, and only possible in case of even number of helices. Whereas the type 4B, we're going to talk about that, has an odd number of transmembrane domains, and the N and C terminus of the protein end up in opposite sides of the ER membrane that we have seen in the previous slide. And then we are going to talk about the GPI anchored proteins as well. And these are some of the examples of these different types of transmembrane proteins or integral proteins. So let's take a look at each type of these separately. First, take a look at the synthesis of the translocation type 1 transmembrane proteins. An example of the type 1 transmembrane protein is an insulin receptor. Its C-terminus is on the cytosolic side of the phospholipid bilayer. Most of the protein and its N-terminus is on the opposite side, and it has a cleavable N-terminus signal sequence. Synthesis of this transmembrane protein, I think, is easy to understand because it is most likely the synthesis of the soluble secretory proteins we just saw. So the initial steps are the same, even though it is not shown in the slide. So synthesis begins with a cytosolic free ribosome, and then the signal sequence becomes exposed, and it is bound to the signal recognition particle, which guides the polyribosomes to the ER membrane by its interaction with the SRP receptor, which is not shown as well. So just remember that SRP and SRP receptors are also involved in this process. Initial steps are the same. The protein then enters the translocon, which is a very large protein complex of proteins, and this is responsible for translocation of the secretory proteins into the ER. Synthesis proceeds, and the N-terminal sequence is then cleaved off by the signal peptidase, and then the rest of the protein is synthesized the same way as the soluble secretory proteins. So as the synthesis proceeds and this protein is being extruded through the pore of the translocon, a string of amino acids will be synthesized as the stop transfer anchor sequence, or the anchoring segment of the protein, or anchoring sequence of the protein. There are many ways you can represent that, and represents the transmembrane region, which is in red, the transmembrane region of this protein. So what happens is that as this portion of this protein is being synthesized, it forms an alpha helical stretch because of it's made up of hydrophobic amino acids. Then what happens is that the transmembrane protein will then diffuse out of the translocon. As you can see, the transmembrane region is now embedded in the phospholipid bilayer. As synthesis proceeds and is completed, what you end up is the type 1 transmembrane protein, where there is a short C-terminal cytosolic phase, and the majority of the protein, or majority of the N-terminus of the protein, it is found in the ER lumen or extracellular space or Golgi. And this is the synthesis of the type 1 transmembrane protein. So what about type 2? So for example, is the transferrin receptor. It has an opposite orientation of type 1. So the N-terminus is on the cytosolic phase and the C-terminus is in the lumen. Again, the initial steps are the same. The synthesis begins on the cytosolic free ribosome, but there is no N-terminal signal sequence. This is the major difference of the type 2 from the type 1. No signal sequence. However, when the signal anchor sequence is synthesized, there is a sequence known as a signal anchor sequence. And this sequence, and again represent the transmembrane region, it itself is bound by a signal recognition particle which guides the polyribosomes to the ER through its interaction with the signal recognition particle receptor. Then the proteins get inserted into the translocon, only this part, this red part. See that this part, nascent polypeptide chain, the N-terminal having some of these positive charge, they are outside or in the cytosol. Only this signal anchor sequence is inserted into the translocon by the SRP, which is not shown in here. And then what happens? There is no terminal sequences to get cleaved off. And now you will notice that the N-terminus is on the cytosolic phase. So how the cell actually know, this is the important part, that how the cell know to insert this protein with the N-terminus on the cytosolic phase as shown. It has been suggested that a string of positively charged amino acids shown here that are oriented just to the N-terminus of the signal anchor sequence. Again, the string of positively charged amino acid is actually regulates the mechanism by which this protein is inserted into the translocon. So once it's inserted into the translocon and protein synthesis proceeds, you get diffusion of the transmembrane region out of the translocon directly into the phospholipid bilayer right here, and the completion of the synthesis. What you end up with is a transmembrane protein with N-terminus on the cytosolic phase. And as you can see, and again, with this, the signal recognition particle is involved, but it is not shown in this process. So the type 3 transmembrane proteins are quite similar to type 1. Their orientation is the same, where the C-terminus is on the cytosolic phase and their N-terminus ends up in the lumen of the ER, but there is no N-terminal signal sequence. It is not known if the signal recognition particle is involved in this process or not. That's why the SRP involved is with a question mark on the lower left-hand side right here. So some unknown mechanism, this protein begins its synthesis on the cytosolic free ribosomes and associated with a translocon in the ER membrane. And in this process, the protein gets inserted into the translocon in this orientation. Again, it is a string of positively charged amino acids, and that are now the C-terminus side of the transmembrane region, which helps to regulate the insertion of the transmembrane protein into the right orientation. And the course and the steps are similar to the previous two transmembrane proteins. The transmembrane region diffuses out of the translocon and inserted into the membrane as well. So the synthesis is completed by the ribosome bound to the messenger RNA, and then you end up with a protein with the C-terminus on the cytosolic surface and the N-terminus in the ER lumen. So this is basically the type 3. Now, this type of protein you can think of as the type 3 in reverse. We call these group of protein as the tail-anchored proteins. The C-terminal anchor proteins pose a challenge as there is no N-terminal signal sequence, first of all, like the many other, to these target these proteins, and the C-terminal signal sequence do not appear unless the translation is completed and released from the ribosome. So all the tail-anchored proteins are made in the cytosol. Or the ribosome is not associated with the endoplasmic reticulum. So a complex of right here is like an SGT2, GET4, and GET5. This picture was not in your booklet, but it's in your text. You can look at the figure legend that this SGT2, GET4, GET5 sequestered the nascent polypeptide and helped it to load onto a GET3 complex, which is complex with an ATP. And the GET3 and ATP complex gets this protein and bring it down into the AR membrane, where it associates with a GET1, GET2 complex. And the hydrolysis of the ATP from the GET3 allows this nascent polypeptide to be inserted into the GET2 and GET1 complex. And this is ADP hydrolysis happens at this point. And from here, this red portion, which is the hydrophobic portion from the GET1-2 complex, diffuse out into the membrane and is inserted into the membrane, the process that we saw previously. And when ADP is released from the GET3 complex, it is again ready to bind an ATP and start this cycle. So this is basically the process by which the tail-anchored proteins work. And many of these large snare proteins found in the yeast, V snare, T snares, they are actually manufactured this way. So the multiple pass transmembrane proteins are obviously much more complicated and a little bit more difficult to understand. But on this slide, it summarizes at least what is known about the synthesis of type 4 transmembrane proteins, which is a multiple pass. In this example, we are looking at the GLUT1 transporter, which passes through the membrane an even number of times, such as that the N-terminus and the C-terminus both end up on the cytosolic side of the membrane. And the signal recognition particle is involved in this particular case as well. So what happens is that the initial steps are the same. The synthesis begins on the cytosolic free ribosome. There is no N-terminal signal sequence for the signal recognition particle to bind, but there's an internal anchor sequence. So SRP binds to that internal anchor signal sequence and directs the polyribosome to the ER right here. So you can see that the SRP signal recognition particle bound to that internal sequence, and it is bound to the SRP receptor. So then the first and the second membrane regions are synthesized and they diffuse out of the translocon into the membrane. And the interesting part is that here's the remainder part of the transmembrane regions are actually synthesized in the cytosol. So you can easily appreciate here is the ribosome that synthesizes the next two transmembrane region, which become inserted into the translocon directly, and then they will diffuse out. And then there will be, again, the next two ones will be made in the cytosol. They will be inserted into the translocon, and then they will diffuse out. And so there must be some type of chaperone protein involved in this process, because remember that these transmembrane domains are completely hydrophobic. So when these transmembrane proteins are being manufactured into the cytosol, it is an unfavorable condition due to the water around them. And to avoid this process, there must be some additional involvement of protein which can mask or cover these proteins until they are inserted into the translocon, so that the hydrophobic interaction can take place and doesn't destabilize the protein into the aqueous environment. So what are these chaperone proteins which help them to bring into the translocon has still not been identified. So this is some intense area of research as well. So those are the transmembrane proteins. What about the lipid anchored proteins? We talked about 3 lipid anchored proteins, and here we will just only focus on the GPI anchored proteins because they are made exclusively in the endoplasmic reticulum on ER bound polyribosomes. So you will remember that the GPI anchored proteins, where the entire polypeptide is one side of the biological membrane and it is anchored to the membrane through an GPI anchor. From the previous chapter, you can review the structure of GPI by going back to the lectures on biomembrane and it's a very complex structure, and this bound to the C terminus of the GPI anchored protein. So here is how it works. It's quite simple. In the initial steps are identical to the initial step of the type 1 transmembrane protein. So here is usually an A-terminal cleavable sequence, not shown in this slide. The protein is synthesized as a type 1 transmembrane protein with a little bit of C terminus on the cytosolic side, right here. And the majority of the protein on the terminus is in the ER lumen. And what happens at this stage, there is a string of amino acid close to the transmembrane region that is recognized by an enzyme known as GPI transamidase. This is not a protein which is a signal peptidase. This protein is important. It's not GPI transamidase. What GPI transamidase does is that it cleaves the protein at that position and makes a covalent attachment with that protein to a GPI anchor right here. So you end up with a GPI anchored protein. So the normal question you can easily ask that why are you going to change the anchor? It is already anchored in the membrane. Why you are going to change the anchor? So the answer is, why bother to change it to a lipid anchor from the protein anchor? As it turns out, the lipid anchored proteins are more mobile in the biological membrane than transmembrane proteins. If you have a protein that needs to be more mobile for its function, if you need it, you need to put a GPI anchor on it. Furthermore, proteins that are GPI anchored have a high affinity for the lipid rafts. If you remember, the lipid rafts are the small domains in biological membranes that are cholesterol and the sphingolipid rich, and it's one of the ways that the cells can confine proteins to a specific domain in the membrane. So if this protein has to function in the context of a lipid raft, then it makes sense to remove the transmembrane region or the protein and replace it with a GPI anchor, which does have a natural affinity towards the lipid rafts. So this is a revisit of the topographic sequence that determines the orientation of the ER membranes. Say, for example, for the type 1 proteins, the N-terminal signal sequence is cleaved, and the single internal stop transfer sequence will be present. This will bring the cytosolic ribosome, polyribosomes to the ER, and the rest of the protein will be synthesized. Type 2 is characterized by a single internal signal anchor or SA sequence. The orientation depends on a high density of the positively charged amino acids on the N-terminal side of the SA sequence, so that the N-terminal is present towards the cytosolic side, whereas the C-terminal is in the lumen. On the type 3, you can see that also contain a single internal signal anchor site sequence, but the orientation depends on the high density of positively charged on the opposite side, on the C-terminal side of the SA sequence. So what happens here, that the N-terminal ends up in the lumen, unlike the type 3, and the C-terminal ends up in the cytosol. So just this crisscross, this was here, this was here. So that small changes are making a difference. So here you can easily see that the type 4A proteins have no N-terminal signal sequence either. Their N-terminus is in the cytosol and have an alternating type 2 SA sequence and STA sequences. That means the high density of the positively charged amino acids are responsible for the N-terminal side of this is going to be towards the cytosolic side. This is membrane anchored, and then it's going to be on the luminal side. Then you have again membrane anchored. This will be the cytosolic side. So you can easily see that wherever you have the cytosolic side, that side of the N-terminal is getting these positive charge and so on. On the other hand, what happens that the last one right here in the type 4B, you have the odd number, and the charges on the cytosolic side end up on the same side of these SA sequences or the internal signal anchor sequences. And that makes both of them go on the different direction. So you can see the N-terminal ends up in the lumen, on the C-terminal is the cytosol, whereas the N-terminal is in the cytosol and the C-terminal is also cytosol, and they have even 1, 2, 3, 4 even number of signal sequences or single internal stop transfer anchor sequences as well. So this is basically the overview of the different hydrophilic stretches, which are in red, whereas the blue are the hydrophilic stretch, where they can either be in the cytosolic side or the luminal side. So let's take a look at the final look at this hydrophobic part. In this figure, we will try to illustrate the concept of those stretches of hydrophobic and hydrophilic profiles. The slide shows what is known as a hydrophobicity plot or hydropathy plot, and that can predict the topogenic sequences in an integral membrane protein. They are generated by plotting the total hydrophobicity of each segment of 20 contiguous amino acid along with the length of the protein. So you can see that positive values are relatively hydrophobic right here, the top portion, whereas the negative values are relatively hydrophilic, or they are more polar. You can see that how you can plot and you can recognize the signal and the stop transfer sequence in the human growth hormone. Here you can see that right here is a signal sequence, hydrophobic, stop transfer sequence, hydrophobic. Here you can see in this one is a signal anchor sequence. On the other hand, this is really remarkable that this is the loop, and you can easily see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 transmembrane helices, and just looking at the hydrophobicity plot, you can tell where these are. So you can appreciate this, that how the hydrophobicity plot just from the sequence of the protein can give you an idea that if it is a secretory soluble protein, it's a transmembrane protein. If it is a transmembrane protein, how many passes it makes to the transmembrane area. All these, is the N-terminal in the cytosol, or is the N-terminal is in the lumen or extracytoplasmic phase. All these information you can derive from the hydropathy or the hydrophobicity profile of a protein. So in these two sections, we have talked about the synthesis and targeting secretory proteins to and across the ER membrane, and also insertion of the integral proteins into the ER membranes to be delivered to different destinations.